right, thank you. So next we have Gabriela LAVEZZARI from PhRMA, who's going to be taking the industry perspective on challenges and opportunities.
Well, thank you, John. Thanks for inviting me. And just for information, we got a strict rules of staying within the 10 minutes. So that's why we are so terrified by the red light. All right. So what I want to talk to you about is an effort that PhRMA-BIO had done in 2014. But prior to this, in 2013, actually at PhRMA, we have looked at the biomarker qualification and did a gap analysis of what are the issues within the industry in moving biomarker in the qualification. And one of those issues was, thanks to Shashi already provided a lot of context for me, is about the context of use in the evidentiary standard. So when we sit down, I think it was a late December afternoon, Bio and Pharma sit down together and thought about maybe doing a survey to really look at what is the industry view on qualification, what are the biomarkers that are important, and then kind of start exploring about the context of use in evidentiary standard, which was kind of a new way for us to really get a little bit more into the weeds of what are the challenges in the qualification. So this effort is actually a first of its kind. We have worked across the two trade associations, and we survey a lot of member company. As I mentioned, collected both the context of use, but also the evidentiary standard. We surveyed 24 companies, 17 from pharma. Many of those pharma companies are also bio member, versus bio had 7. So pharma approached the large size company and bio approached more the mid-size and small biotech. We have 3 different modules, and I'll go through that. But we had a very good adherence in the non-clinical safety. We have 90% compliance, and then 75 for the clinical safety and 79 on the efficacy. So this is the module and the question that we asked. Initially, we asked for the non-clinical and clinical safety and non-clinical efficacy. We asked about the complexity. So thinking about biomarker qualifications, specific organ or therapeutic area, what are the scientific, technical, and regulatory complexity? And we define those, what we mean by that. We then asked the question of how, if a biomarker could be qualified, would increase the efficiency in drug development. And then specifically, we ask what would be the best context of use that would be most useful for drug development. And based on that context of use, what in their opinion would be the evidentiary standard needed to support the qualification package. And then as option, we asked to actually provide a rationale why the array of evidentiary standard would be sufficient, and also what would be the biomarker of choice in that specific topic. As I mentioned, we have both clinical and non-clinical safety, and we look across different systems and organs. And then specific for the clinical efficacy, we looked at the therapeutic area that actually mirror the FDA organization under review. When we talked about context of use, I hope you can see a little bit. We have provided some example, but also we have provided an option to write exactly what the company would think about. And then we propose a variety of different evidentiary standard example. So surveys are, difficult tools. They're great tools to gather information, but they are very tricky and provide definitely a lot of bias. So we were able to collect a lot of data points. It was a very big survey, and we need to have a tool that could help us to kind of see through the noise and see if there was actually something that was interesting. So what we did, we compile and create a score. So we look at the complexity, both the scientific, technical, and regulatory, and collapse those in one score. And then we graph that against the efficiency in drug development. We actually play about the complexity and taking one out and see if the score would change. And we saw there was no changes in that. And then we created this kind of quadrant of what that means. So you have the low left quadrant, that is low complexity and low efficiency, where really there is no benefit, to what will be a transformational, where will be a lot of efficiency in drug development, but also a lot of complexities. So these are the snapshot and the distribution of the non-clinical safety biomarker. Everything falls in the top right quadrant. But what was interesting for us is kind of a negative control was the hematologic toxicity. So a lot of the members responded mentioning that what we have currently has biomarker in the hematox are sufficient. There is no really need to qualify more. So it was a nice point for us to really validate the survey itself. In the pancreas toxicity, actually was interesting. A variety of members mentioned the difficulty in working in that area, also where the science is not there yet. So an area where there was not a good consensus of what could be done in that space. What I thought was a strong consensus was definitely the nephrotox and the hepatotoxicity. This moves to the clinical safety in the biomarker. Again, the pancreatox is kind of an outlier. Again, not much consensus, and still the science is not there yet. But then the usual subset in the hepatotox and nephrotox, and definitely some interest in the cardiotoxicity. So then I mentioned that we ask about the contexts of use. So what we did here in this graph, we picked what would be one specific context of use, and this is the demonstration of toxicity without histopathology, and ask what would be the evidentiary standard to support that. And what we have done as analysis is to actually look across different organs, still in the acute safety between cardio and nephro. What was any specific pattern on the evidentiary standard selected? And what we saw